All right. Well, good morning, everybody, for Thursday. I am Katie Barker. I run the Secretariat Office for the Global Genome Biodiversity Network from the National Museum of Natural History Smithsonian Institution. The co-organizers of this session are also Gabby Druga from the Berlin Botanic Garden. She's the technical manager for GGBN, and Jonas Astrin from the Leibniz Institute of Biodiversity Change. He is the executive committee chair for GGBN. So basically what I wanted to do for this session was come here and really talk a little bit about GGBN's best practices and policies for running and operating genomic biobanks, mostly biodiversity genomic biobanks, natural history collections, zoological collections, anything that's not human. So I'm going to start off, I'm going to do a brief overview of GGBN, and then we're going to move into some examples of policies and practices guidance that's been developed through the Synthesis Plus project. So there'll be 3 talks there. And then the last talk will be given by Gabby and Anton on the GGBN data portal and updates to the portal. And then we'll have like 15 minutes at the end for discussion and questions. So I will go ahead and get started. So why did we even start GGBN? The whole reason we started networking genomic collections across natural history museums was because there was a need for scientists to have access to samples. They didn't know where they were. There was no information about where they could get them or how they could use them. And we also didn't know what the gaps were in the tree of life. So we didn't know what the gaps were in the collections themselves. So we needed to network these collections and figure out what the gaps were, what was available, and how they could be used. And of course, the scientists themselves need to be able to guide future efforts in strategic sampling to fill these gaps. So GGBN meets the needs of the research community by making genomic collections discoverable for research through a network community of biodiversity repositories. What we're doing is we're basically bridging the gap between source material, so the vouchers and occurrence data in GBIF, with the molecular data in NCBI and other related databases. And we're doing that by linking all that information to the DNA and tissue samples. So GGBN was started in 2012 by 13 natural history and biodiversity informatics institutions. And since then, we've grown to 112 members across 38 countries. We have over 6 million records online. This is all biodiversity information, plants, fungi, animals, microorganisms. And we have over 97,000 species represented within these collections. So what are the benefits to research and collections? GGBN provides one-stop access to well-documented samples, vouchers, provenance information, quality control and assessment of those samples. As I said, we're providing the standard information for over 97,000 species currently, and we're growing steadily as new members join and provide their collections online. Right now, we have about 38 members who are providing data to the GGBN data portal. GGBN also provides the opportunity for knowledge exchange, so researchers can work with the collections, the GGBN members, and basically use their research questions to help drive the growth of these collections. For collections, we provide visibility and impact for those genetic collections themselves. We provide best practices for managing those biobanks, standardized vocabulary for publishing those data online. We're providing standard procedures, for example, preservation, quality control, and quality analysis. We are linking to those aggregators, GBIF and INSDC. We also provide technical support for organizing those collections and publishing them online. And of course, very importantly, all of our members maintain ownership of their samples. So we provide, like I said, we provide best practice guidelines for the GGBN biorepositories, and we're basically supporting the biobank operations of these members. And basically what these members are doing is they're providing genetic samples for research use, and we provide all the guidance and infrastructure to help them do that efficiently. We have 2 guidance documents. We have a code of conduct and a best practice. These were developed in collaboration with CTAF and Spnhc. So our code of conduct is governing principles under which our collections are managed and collections-based research is conducted by our members. And very importantly, we have a use of materials statement, which explains how specimens are used by GGBN members. So if you are providing the tissues to the biobanks, this use of samples statement, or sorry, use of materials statement is really important because it provides transparency to those providing countries. The best practice provides details for implementation of the principles of the Code of Conduct. So in addition, we have a GGBN data standard, which was developed, I think, at a Tdwg meeting back in 2012. And this is an official Tdwg standard. It complements Darwin Core, ABCD, and Mixis. And it really allows for efficient sharing and use of the genetic samples across GGBN member biorepositories. We have controlled vocabulary for tissues and DNAs. There are 9 mandatory fields and 39 recommended fields. And this is implemented by Arctos, Specify, and Symbiota. And we're currently working with Species 360 to implement this with ZIMS, which is a zoological information management system. Oh, and I had the link was there, so if you guys are interested in looking at our wiki page, and you can look at the standard and see all the details about those recommended and mandatory fields. So in addition, we have permit vocabulary. So the whole reason we're doing permit vocabulary is because we want to provide information about where these samples came from and how researchers can use these samples. So this is really providing transparency across the GGBN members about how they're providing these samples for use for research. These permit fields support the compliance with the Nagoya Protocol for access and benefit sharing. And the permits themselves, they're linked to the samples, but the actual text of the permit is not available online for privacy reasons. But if you want to know about it, the researcher goes to the biobank, and the biobank provides you with that information. So we have 4 examples of GGBN publications. The one on the top left is our data standard specification. It's available on our website and online, so you can read all about the data standard that was developed at a Beijing, I think, Tdwg meeting. We also have a publication on the data portal and a couple of publications on various tools that we've developed over the years. So our priorities for 2024, we're really working to explore partnerships and to leverage the partnerships that we already have with respect to biobanking and genetic resources. So we're working with ISBER, IBOL Europe, ERGA, Spnhc, ENA, Species 360, and BOLD. And we're also working to complete GGBN stable identifiers to enable cross-links with other platforms such as ENA, GBIF, and DISCO. And I think Gabby might be talking about this a little bit later on today or during the session. And we're also rolling out the new GGBN webpage to increase visibility and improve accessibility of our collections, which Gabby will definitely be talking about later on. We are also starting a barcoding project test case, which makes EU bioscan samples from non-GGBN members available in GGBN. And we're going to continue to improve community communications and increase awareness of GGBN and its resources so we can build the membership, get those collections online, and make them discoverable for research to the research community. We're also currently supporting a Horizon scan on environmental sample data and management. And that's being run through the Smithsonian with my colleagues at Natural History. And we're also holding annual or biannual GGBN information meetings with Q&A to increase awareness of GGBN benefits and activities. So I'm sorry, my slides are a little screwed up, but hey, it's all good. So our recent community activities, we had our last conference in October of 2023, hosted by the University of Autónoma de Aguas Calientes in Mexico. And in April, we had 2 virtual workshops on access and benefit sharing and digital sequence information, which were co-hosted by our colleagues at ERGA and the Earth Biogenome Project. And those are also recorded and are on our YouTube channel. So upcoming activities include November 13th. We're going to have our first virtual information session, so anyone who's interested is very welcome. So just feel free to reach out to me. And then the first week of October 2025, we'll be holding our next GGBN conference in Cape Town, South Africa, which will be hosted by the South African National Biodiversity Institute and Biodiversity Biobank South Africa. And this meeting will be in conjunction with ISBER. So ISBER will be having parallel sessions at this meeting related to biobanking. And ISBER is a more human biobanking network, but they have relevant best practices related to how you operate a biobank. So just to conclude, so what are the benefits of networking genetic collections? This is increased efficiencies. So if you network the collections, you are basically reducing the amount of work that one institution or one biobank has to do because you're sharing standards, sharing best practices, sharing resources. We are building on existing best practices and standards, so we're developing collectively, sharing knowledge to build collectively collections and data management systems, understanding and abiding by laws and regulations such as Nagoya, and that's all outlined in our best practices, and working together to develop sample quality assessment and quality control. So if I want to do a metagenome, I know that this sample is good enough to do a metagenome or a whole genome. And of course, what happens is that when we network our collections, we basically have greater impact and visibility of these collections, which is really important for fundraising and just institutional support for these collections. So, thank you. All right, so I was quick. So we have about 2 minutes left if there are any questions about GGBN specifically. You all got it? It makes perfect sense? Cool. All right, well, I'm gonna sit on it for a minute because we'll stay on schedule. Yeah, please.
I just want to remind everybody that online people can post questions in the chat in Zoom now, but once the session is over, you can still interact with speakers and other session participants in the chat in Oxford Abstracts. There will be a group chat for this session.
Thanks, Stan. Come on down.
Hi. Thank you. You mentioned that, sorry, my name is Henard from Leibniz Institute. You mentioned that the permits, the codes of permits are not shown in the publicly in the database. But I wonder if users that upload the material if they want to show the permits, can they do that, or is that possible?
So what we're doing is, so the permit numbers are actually, they show up in the, on the page for the sample itself on the GGBN data portal. So you can see the number, and then it basically provides information like metadata on what those permits are for and what the general restrictions are. But if you actually want to look at the full permit, you have to reach out to the biobank manager, and then they provide you with the information for the full permit.
Excellent. That's it. Thank you.
So transparency, but not violating privacy issues. So awesome. All right, thank you. Do you want to pull up the next talk? All right, so our next talk is going to be from Carolina Corrales and Jonas Astrin. Carolina is presenting remotely today, and this is our first of the 3 Synthesis Plus talks that reflect the best practices and standards that GGBN is providing to the community. So this one is the release of a handbook on biodiversity biobanking practices.
Good morning, everyone. I'm Carolina Corrales, and I, together with Jonas, are 2 of the authors of the handbook on biodiversity biobanking practices that was developed within the Synthesis project. So first of all, why is biodiversity biobanking important? Biobanks are one of the crucial tools to safeguard our planet's biodiversity along with preserving its genetic diversity. Biobanks have had an important role in crop and livestock improvement, also in the development of medicines and industrial products, and more recently, in the conservation of genetic rescue of threatened species. However, not all biobank communities have standardized procedures or are currently developing their own standards. Most of them follow the iceberg guidelines, but this doesn't contain specialized information regarding culture collections, seed banking, or environmental samples, and most of its content is focused exclusively on human biobanks. Therefore, the Synthesis project had 2 main goals regarding biobanking. First, to develop a landscape analysis among biodiversity and environmental biobanks to identify common practices, gaps, and challenges. And second, to develop and disseminate best practices to support biodiversity biobanking activities. For the landscape analysis, we conducted a survey, contacted other institutions worldwide to have access to their standard operating procedures and protocols. We also carried out a vast literature review to find similarities and gaps along the biobanking workflow for all types of biodiversity biobanks. This means that we got information on every single step of the biobank workflow, starting with fieldwork practices, first preservation and transportation to the lab, culture procedures, DNA instructions, preservation storage methods, retrieval of samples from preservation, use of samples by others, meaning loans or publications, and how the metadata for every single of these steps was collected and stored in databases. All those previous steps were revised for all organisms and samples listed here. We did not include viruses, bacteria, and humans because they are all considered extensively in other guidelines. So after checking the survey's answers, contacted curators and biobank managers, and read tons of papers, we found 8 crucial gaps that affected all biodiversity biobanks. First, at large institutions, protocols are not standardized as each section or department develops its own. This is often related to the lack of a centralized facility within the institution. Then, researchers working with microorganisms, plant-associated taxa, and marine samples tend not to deposit specimen vouchers or molecular vouchers at specialized repositories, so it is not possible to further study those organisms and the reproducibility principle cannot be applied. Moreover, backups and duplications of collections are still uncommon. There are also issues with digitization of collections due to the lack of human and financial resources. And last but not least, permits, transactions, and publications associated to the samples are not traceable. Hence, it is not possible to assess the collection's impact in research. The outcome for the second goal was the handbook. Here, we compiled all commonalities and gave possible solutions to challenges and ways to fill in the gaps. It consists of 12 chapters with 2 big sections, one related to viable cells and the other one to DNA. All chapters are arranged by organism or type of sample. It is important to note that the handbook is more a compilation of protocols and does not explain in too much detail every single of these protocols. It has, however, a very large reference section organized by topics. It also includes a list of all biobank facilities we encountered in our search. We also would like to highlight that one of the main tasks of biobanks is to preserve viable material, as it is not only a source of high-quality DNA, but also a renewable resource that can contribute to conservation projects. Cryopreservation is the gold standard for preserving these samples. However, it is not yet possible to cryopreserve all species, as there is still a lack of knowledge about the physiological requirements for most of them. It is also important to note that a universal protocol doesn't exist. Hence, protocols must be carefully adapted to each taxon and cell type to maximize the potential of cryobanking. In the case of natural history museums, we highly recommend to start applying the extended specimen network concept, which means that a voucher should include extensions or associated data, like for instance, images, metadata, molecular data, DNA and tissue, and morphometric information. Environmental samples and viable cell cultures can also be collected along the process. All this information should be included in a database to be able to track any addition or change to the specimen data. Natural history museums, apart of having dry and wet collections, should also include biobanks within their facilities to store those associated samples that cannot be conventionally stored and to better preserve the integrity and quality of these samples. To conclude, we consider our handbook a first step towards the standardization of biodiversity biobanking practices. Still, it is crucial to remember that because of all the different kinds of living organisms and cell types, not all processes can be standardized. Most procedures, however, rely on preservation protocols, but the handbook also offers other alternatives when cryopreservation is not an option. You just saw my child passing by. And to finish, I would like to thank all contributors who made possible to develop this handbook and so many others that are listed on our acknowledgement section. And of course, thanks to you for listening.
Thanks, Carolina, who's not actually here. Sleeping probably in Europe, I assume. So they're not here to answer questions, but do you guys, are there any questions that I can attempt to answer? Or Juta might be able to help. Thank you, Juta.
Hi, I'm quite new to GGBN, I have to admit. I'm wondering, are you, like looking into the future, are you thinking about creating one central facility, or do you want to have kind of like a distributed, currently I think it's a distributed network. Do you want to keep it that way? What about kind of like mirroring your repository so that samples are kept safe?
That's a really good question. So the idea of GGBN is that it's distributed collections, but there's one central data portal, which is hosted by the Berlin Botanic Garden in Berlin, Germany. But the idea is that we also, part of our best practices that I was talking about and the recommendations are that we do mirror collections. So it's like an age-old best practice, right? Because if one collection has all the microorganisms for a specific species in the world and that goes down because of a natural disaster, we want to make sure that there's parallel collections in another GGBN institution. So that's kind of one of our goals as we're moving forward, and what we're talking about with respect to best practices is how do we ensure that the GGBN collections are actually mirrored, because they're never going to be in one central facility, right? They're going to be dispersed all over the globe, and we want it to be dispersed all over the globe, because another important thing is the provenance information, and that, you know, we have samples from Brazil and South Africa and Colombia and Vietnam, and we want those samples to stay in those countries. And that's actually a really important part of GGBN, is really making sure that like we're supporting the institutions in these countries and helping them to set up their standards and best practices and set up their biobanks and support them so that they can actually do that, and then still link the information to the data portal so that they're available for research. Another thing that I wanted to say about GGBN samples being available for research, if a GGBN member institution publishes the data, that doesn't necessarily mean that they have to share those samples. It just means that they're here, they're protected, they're well preserved, this is the quality, but there might be restrictions associated with those samples that, you know, you just can't share them, or the institution just doesn't want to share them, and that's fine. It's more important to have the samples discoverable, which really helps, I think, in funding for the institutions in the long run and helps the research community. So that was a long-winded answer, I apologize. I guess we'll take a pause for 2 minutes so we keep up with the group. Thanks, Dan.
I have a quick question. Dan Mulcahy from MF in Berlin. On your talk, Katie, I noticed a gap in the map here in Japan.
Yes.
Are there any prospects? Are there biobank collections here that you've been
No, we have, well, there might be, but
Yeah.
that's a really good observation, because that's one country that we have not found a member institution for. So I'm confident that there are tissue and DNA collections that are of a natural history basis in this country. And so we just need to figure out who the contacts are and reach out to them. So, yep. We are in discussions with DDBJ, but yeah, aside from DDBJ, which is an informatics organization, we haven't figured out who the biobankers are yet. So if you guys know of any Japanese biobankers that would like to participate in GGBN, we're very happy to welcome them. Stan, do you want me to wait 5 minutes until I go to the next one?
I think you can go ahead.
Okay.
Just because I think we can keep the discussion going.
Yeah.
But, put up another talk, more to discuss.
Okay, sounds good. Do you want to put up the next talk? All right. So this is our second talk that's focusing on the Synthesis Plus project, which is the EU-funded project that many GGBN members did a lot of work on. And so Edmund is going to start us off, but we have a couple of speakers, and Jutta is one of the speakers on this, and she's in the audience, so she will happily help to field questions. And Dan is also an author, so. All right.
Ladies and gentlemen, thank you for joining us for our presentation on tools for fulfilling legal requirements of biodiversity specimens. The context for our work is the increasing number of laws, permits, contracts and agreements governing the work with and the life cycle of biodiversity specimen. This includes collecting, storing, borrowing, as well as subsequent research, occasionally complemented with developing new products. At present, the legal requirements are quite time-consuming, and an easier management would be appreciated by many. Because often, legal texts from dozens of different jurisdictions are involved, useful improvements would be standardization of terms and processes, international harmonization of key policies, and a comprehensive collection management. Any implementation of tools providing access to legal information and associated functionality is resource intense and will require a dedicated IT infrastructure workforce. In the European project Synthesis Plus, a task group was formed to build on the GGBN data standard and GGBN vocabularies. We worked to improve the GGBN permit vocabulary and the GGBN loan vocabulary. Members of the task group and authors of the resulting publication work on 3 continents at CETAF member institutions and GGBN partners. We wanted to create a comprehensive and uniform classification scheme for documents, compile typical terms that can be found within these documents, and create an exemplar framework for automated processes facilitating swift decisions on a large scale. In the beginning, we only had 2 targets, and you see them here as number 1 and number 3. I will explain on the next slide why we had to add target 2. The main outcomes of this work are published in a 2024 report in the journal Research Ideas and Outcomes. The biodiversity permit contract typology is the classification scheme for legal and contractual documents. The classification is based on the specific purpose of a given document and includes 7 document categories and 38 document types. When looking at the contents of documents from different countries, we realized that even documents with similar purposes may contain very different terms when issued by different countries. Look at the colored lines, where each color represents a specific country. While countries may use the same or at least similar terms, you can see that they do not place them in the same document type. Hence, knowing the document type may not provide sufficient information on what is allowed, required, or restricted. That was quite a setback. But then we created the typology of legal and contractual terms with a preliminary set of 87 terms, which makes it obvious what is allowed, required, or restricted. This allows automation of processes for swift decisions on large scale. The advantage of our two typologies is their flexibility. You can choose whether you use only document categories, only document types, only terms, or a mixture of these three options. The next slides will present examples for categories, types, and terms, so please remember the relationship shown here. This is an example for a document category and its definition. It can be quite difficult to find definitions that are valid across countries. On the right side, you see a colored table with all document categories and document types. It is accessible via the QR code. Our publication has only a black and white table. Here, you see an example for a document type and the relevant definition. The advantage of our group is that we live in a variety of jurisdictions and additionally know legal and contractual documents from many more jurisdictions. This gives us the opportunity to find definitions that fit this wide range of jurisdictions. The typology of legal contractual terms contains an initial set of 87 catchphrases. These are short phrases describing the essence of a specific term in a document. The number of 87 is based on the limited resources available in our project. It is foreseeable that more will be necessary in future. The typology also contains information on the lawful source and whether the catchphrase refers only to the past or to the future, information that is especially important to support automated processes. You see an example for a catchphrase: free further use with reporting requirements. Each catchphrase comes with information on possible lawful sources, for example, documents, laws, or contracts and deeds, a definition of the contents, and whether it applies to the past, the future, or both. Harvard's Museum of Comparative Zoology has implemented this typology in their collection management system. Rita has the details for us.
Thank you, Edmund. MCZBase is an independent instance of the Arctos platform. The Permissions and Rights module allows users to create digital records of permits, agreements, and other legal documents. In addition to adding information about the people and dates associated with the document, a drop-down list can be used to assign a specific document type. Given that specific document type, a document category is automatically assigned. PDFs can also be uploaded and associated with the document. Once created, permissions and rights records can be associated with transactions such as accessions and loans. When a record is added to an accession, the documents are automatically associated with all specimens that have been included in that accession. Similarly, it allows automatic association of documents if accession specimens are added to a loan. Lastly, permissions and rights records can be added to a specific loan shipment, since there are often additional permits needed to transport loans. When adding information about the permissions and rights document, there is a text field where users can add information regarding use restrictions, negotiated benefits, and any benefits provided. If specimens with restrictions are added to a loan transaction, a box similar to the one that you see here in red appears, informing staff that they should review these details before moving forward with the loan. MCZBase is linking legal and contractual documents to collection objects and categorizing them using the permit contract topology. I will now pass it on to Jutta.
Thank you, Breda. The terms topology will enable providers to go one step further, making governance information machine actionable. Rights management modules within collection management systems can automatically compute and arrive at decision proposals. These proposals can support collection staff in efficiently evaluating associated rights and drawing conclusions. The proposals will always require the review by collection staff, who carry the responsibility for the final decision and action. The developed exemplar decision model is based on the W3C provenance and the W3C Open Digital Rights language ontologies. It considers and combines information about the physical entity, involved agent and their roles, the activity in question, for example, a loan transaction, and conditions codified in the form of the typology of terms. The ODRL commands might look like this code block in a JSON linked data serialization. Encoded are two rules that apply to a specific specimen in a loan transaction between two institutions. In this case, the specimen can be shipped only to institutions within the EU, while physical analysis on the specimen are always allowed. Thus, instead of going through the documents again and again, the system will summarize the applicable policies and propose a way forward. I will now pass back to Edmund.
Thank you, Jutta. In our opinion, the next step should be, first, the development of the two typologies into an internationally recognized standard. This involves setting up a dedicated Tdwg task group in charge of preparing and transversing the ratification process. Second, we propose that a survey could inform, guide, and accelerate the implementation phase by gathering information on already existing ways of data processing for permits and terms, and by identifying impediments to implementation. Third, simultaneously GGBN and interested CMSs can extend the existing rights management modules by adding the developed typologies and functionality enabling further automation. In this way, they will gather experience and create worked examples that can be shared with colleagues. We call the community to action on integrating digitally supported functionality for the governance of collection objects and biodiversity data into their collection management processes and data infrastructures. Let me thank all my colleagues in this team, and let me thank you for your attention.
Thank you. And Edmund, yep. I think Edmund, you're online.
Yes, I am. Can you hear me?
Lovely. Yes, we can. Lovely.
Thank you.
Thank you for staying up so late. We appreciate it.
I got up again.
So any questions for our lovely authors, Edmund, Jutta, Brita, and Dan? Yes, Anton. Yeah, the mic.
Thank you. Anton Günsch from the Botanic Garden, Berlin. I wonder, Edmund, you said on the last slide that you would like to see some kind of Tdwg standardization process for the vocabulary. I wonder, I mean, we all know that Tdwg standardization takes time, often years, and whether such a process would kind of hinder the implementation process because people would wait for the standardization to be completed. Shouldn't we do it the other way around, start from here, because I think the vocabulary is quite mature, start from here, create several implementations and create best practices from them, and then go into the Tdwg standardization process.
Well,
Dan has
of course you're right, but it depends on what the community wants. At present, we had a small team developing these typologies. And the community might have the opinion that they are not ready for implementation yet. But on the other side, you're right. Standardization takes time and people have to wait. So it's up to the community how they want to start.
Stan Bloom, just commenting on the process. If you already have a standard in place, then you're in the maintenance mode. And maintenance of a standard is easier than getting a new standard adopted. So you just basically need to propose the changes, have a nominal period of public review, and summarize the impact or the amount of comments got received on the standard during public review, and it can be approved. It's a much lighter process than expert review and an open public review.
You are right, but as far as I know, and I'm not the expert on that, the GGBN vocabularies formally are not a standard yet.
I think where are we with respect to the ratification process? I think we're pretty, I mean, I think it's a formal standard, right, with Tdwg, it's just not fully ratified. Is that correct, Stan or Anton? Who gets there first?
It's in a kind of a problem situation, I guess, because it was approved in some sense before the current documentation specification was approved. So you're kind of not compliant. We would very much like to get the documentation into the form that is current. And maybe the new convener of the GGBN interest group can make that happen. But yeah, there's work to be done there too.
Anton, did you have a comment?
Sorry, use the microphone just to say no, I don't have a comment, because what Stan says is, yeah, it's a situation. We have to put some work into the documentation. Yeah. Yeah.
Hi, I'm Jessie Nakano. I'm at the Smithsonian, the Paleo Department, and I'm a registrar there. I think this is really great. It's something that I know our system has, like the setup in terms of rights, and like I don't think we have as updated of a standard though. So I think more conversations and seeing what that is and how we can implement it would be really great. But I think this was really great to hear. Thank you.
Thank you.
Any questions online, Stan? Okay. Oh. All right, do we want to move to the next talk? Thank you, Edmund. We appreciate it.
Welcome.
All right, so we're going to move to our third synthesis project talk. And this is Policies for Molecular Collections by Astrid Demestier, and she's also remote.
I'm Astrid Demestier. I work in the Botanical Garden in the Freie Universität. I'm a postdoc there. And I'm going to present to you some policies for molecular collection, and especially the handbook that was published in the beginning of the year. So molecular collection, you all know what it is. It's any kind of sample that has the capacity to store genetic material. So here I'm going to talk about the molecular collection of tissue sample, cell culture, environmental sample, and DNA. So they are a key tool for scientists. I guess you all know that. They are growing worldwide. We've seen that in the past few days, but they're growing very fast and faster than the guidelines that are. So one key point of molecular collection is that it makes sense if it's available for everyone. It makes sense if you can exchange them. So for that, you can go to institutional database and look for samples that of interest you, or you can go to existing platform like GGBN. But all of this process of exchanging material between institutions goes with a lot of regulation and policies, and that can be very hard to find your way through it. So it goes with a precise follow-up of the sample, and you need to comply with regulation. And so as part of the Synthesis Plus project, which was a project that brings all European institutions together to discuss the management of natural collection, and so that included molecular collection. And so one of the work package was about writing a handbook to help you, show you guidelines how to best manage the molecular collection. And that gave birth to a publication at the beginning of the year, which is the Policy Handbook on Using Molecular Collection. So it's a huge project that was made with 28 authors from 13 institutions, and it's really focused on, it aims, well, it's available right now in open access in the RIO website and in English. It's for researchers and institutions, and it really wants to provide optimized workflow and standardized workflow to best manage molecular collection. So I'm going to present a bit the content of the handbook, but it's 5 chapters, one about receiving molecular sample requests, the second one about requesting molecular material to other institutions, The third one is about sequence submission. The fourth about managing all the legal information associated to the sample. And the fifth one about an AS legal obligation. And I'm not going to present it, but I'm just pointing out that there is also a very important part on useful resources. So the first one is receiving a loan request. So basically you're an institution and you have a molecular collection and someone from another institution is asking you for one of your samples. So you have a loan request that comes in and then what do you need to do? So we really go through every step to explain how it is best done. So really document the request, track every information related to it, and this is better done by permanent staff. So we are sure that the request doesn't get lost if the researcher go away. And obviously the establishment of a material transfer agreement that goes with the loan that will really define the use of the material, if it can be transferred to third parties, can it be used for molecular combination use, and what do we do with the remaining from the product? Can it be kept at the institution or does it have to come back? So, and then after signing your MTA as the molecular collection, you can just prepare and ship the loan, ask for an acknowledgement that the loan has been received. And then when there is completion of a research project, we need to clarify if the loan needs to come back to the original institution or if it can be kept in the other one. So in the handbook, there is a really cool diagram that I really motivate you to look at. We try to really describe the workflow and show every stage and when you need to document things and what are the documents you need to have at every step of the way. Then the second part of the book is about requesting a loan. So that time you are an institution or a molecular collection and you want a loan for your research. So basically you have a research project, you search in a database like GGBN and you find something on interest and then you send a request form. So what do you need to take care of and what do you need to really pay attention to? So obviously you send the request, you make sure that your MTA is signed. You cannot accept anything if you haven't had like the MTA. And then you need to properly manage the material once it's arrived. So put it in the freezer if it needs to, or properly store it and notify the institution where the project is finished. And as for the first one, we use the same template to do also a diagram with a described workflow, really showing you what are the different stages and what are the documents you need to have. The third chapter, I'm not going to elaborate on it, but if you're interested, really go and have a look. It's about the sequence submission. So basically, when you do a research project and you extract DNA and then you amplify that DNA for central sequencing of phylogenomics, it's usually go into a database. Gene bank, INER, or Tdwg. And so this is a process that can be a bit complicated, so we go through the different steps in that chapter. Now the fourth one is really managing all the legal information, the rights and restrictions that are related to that sample. So this can be quite tricky to really keep track of everything you need to have, like all the permits associated to your sample, but no permits, no samples. You cannot really use something if you're not sure that everything was collected properly and it was acquired properly and so on. So basically in that chapter, we go through all of the different legal information to try to clarify everything and really note like which one are the ones that are really important. So there is fourth information that are related to your sample normally. It's the intellectual property rights, the legislation related to international convention, the CITES for protected species, and the Nagoya for the protocol for access and benefit sharing, that I think we're going to have a lot of talk about tomorrow. And then the rights with the local legislation that also needs to be taken into account. And in case of human material, there is always the issue of consent. So we really emphasize about the fact that you need to always have a digital record associated to your sample. So a sample is not worth, you really need to have all the information that are the collection data and also the commercial permits associated with it. So you need to keep track, to have a database for that to really keep trace of this information. Now you need to have obviously the material transfer agreement associated to it that we already discussed, that specify the term and condition for use. Can you then like lend it to some other institution if it's a tissue material and you only use some part of it? Do you have to give it back? Like what's going to happen? And all of this is in the MTA. And we really emphasize also in the importance of establishing standards. So don't reinvent the wheel every time you have a loan that comes in or the loans that comes out. So really have a standardized workflow so people really know what to do and all of the steps. And this is what we provide. It's not a law, it's really like guidelines and yeah, what we think it will make things better and the process easier. because the properties of molecular collection is good for everyone. So it's beneficial for a researcher, from the other institution, and it's really the point of having molecular connection that can be used upon. And we really like workflow in that handbook, so we have 4 more workflows regarding this legal information. So really to help you go around and really see what are the different things. And I'm not going to go through it, it's quite complicated, but yeah, I think it's really important that to look at it, and it's supposed to be really made for helping molecular connection to be best managed. So let's go to right, and then the 5th and last chapter, it's about compliance with ABS and legal regulations. So it goes a bit more in depth into all of this international convention that was made, the CITES in particular, and Nagoya Protocol, to explain to you what does that mean for you in terms of your sample and acquisition and the permits that are associated to. So we also, as usual, say that it needs to be well documented. You need to have it associated. You cannot have a sample and then lose the information related to it. So really find a way to connect all of them. And then the Nagoya Protocol that is also very useful between questions, especially in between countries. And that's it. So it was really made to establish guidelines. It's about proposing workflow, making things easier, making things better to have like really a proper managed molecular connection and don't have to reinvent it every time you do a new molecular collection. And to emphasize about how important it is to standardize the way molecular collection are handled in laboratories. And that's it. So I'd like to thank everyone that was in the work package that made this possible.
All right, thank you, Astrid, who's also sleeping in France, probably somewhere. Okay, so any questions that have come up? Yes.
Thank you for the presentation. I would like to know what type of information is collected after the loan has been given or finalized. Is reports or what kind of information is coming back to the collections after the loan? Thank you.
Dan or Yuta, can you guys help with that?
That's Dan from MFN again, Berlin. That's a good question, and often on loan contracts, you can put that kind of information, such as, any taxonomic changes made on the collection specimens, and most importantly, if data are published, a link to those, like GenBank numbers, accession numbers, bold numbers, using the proper collection codes, saying we want it cited this way, use this institution code, this collection code. Now you can put all of this in the contract, whether it's followed or not by the users is often not, but that's what we strive for. So.
Hi, Bill Moser at the Smithsonian. Although the United States is not a signatory to the Nagoya Protocol, most major institutions do follow it, so it complicates matters a lot. And I was wondering, in practice, boots on the ground practice, it's very difficult to do third-party loans for the collection staff. It adds a risk to the institution, and have people looked into having the loan recipient sign an MTA with the source country?
Anyone want to take a stab at that?
Actually, that is something I wanted to, it's kind of like going in the same direction. So Jutta Buschbaum here, posing this comment or question as I'm the representative to the UN Convention on Biological Diversity for Spnhc. And so I have been attending the sessions of the convention's bodies for 2 years. And so gotten a bit into the science policy process. And I was wondering, anything that is decided, and currently there's this major negotiation for digital sequence information, which would have or will have a major impact on our community and on the goals and objectives of GGBN. Wouldn't it make sense to become much more actively engaged in the process? What are the thoughts of GGBN about kind of like advocating for an environment that would make these problems easier for our community?
That's a really good question. So GGBN is represented in all the COP meetings. We have representation there from the Natural History Museum of Denmark. And also the DSI network are the chair. We have a policies task force, and Amber Schultz from DSMZ in Germany is also representing GGBN during those discussions. But yeah, so we're in those discussions, listening and advocating for GGBN. But yeah, and we're trying to communicate all that back to the membership and make sure that our interests are advocated at these forums. I love it. Keep going.
Yeah. So I have the feeling maybe there needs to be more discussions because the last policy brief I saw that went to the EU, I had discussions with colleagues where we were rather worried about what kind of direction it was taking. So maybe the different communities in this area who are interested in science policy should communicate more or get more together and try to find like an integrated approach.
Yeah, no, I think that's a really good idea. And we're actually in the middle of the process of renewing our agreement with Spnhc for an MOU for our collaboration. And so I feel like that's one of the things that we should call out and figure out like how best can we join forces and work together to advocate for the interests that are related to both of our organizations. So that's a really good idea. So maybe I'll be in touch with that specifically. So. Okay, next. So we're gonna move on to the last talk. This is by Gabby Druga, who's our technical manager, and Anton Gunst. And this is the new GGBN data portal and GGBN stable identifiers.
Good morning, everybody. My name is Gabi Dröge. I'm the head of the GGBN Technical Secretariat located at the Botanic Garden in Berlin. And I'd like to tell you a little bit about the new GGBN data portal that is currently under development and the stable identifiers. So this is our new homepage. And as you see, it's quite different than the old one. You will have a main search bar where you can search for all 4 resources that we have in GGBN, which are the sample, specimen, cultures, etc. big database with 6.6 million material entities. The other one is the document library with over 1,000 documents. Then we have a big collection of wiki pages with Q&A, ABS information, etc. And last but not least, our member registry with our 112 members. So you can access this page already. It's devportal.ggbn.org. But keep in mind that it's still under development, so some things might not work yet. So this is how the search overview page looks like. You see the results from all the 4 resources, the samples, the member registry, other information, which is the wiki and the library. And then you can go deeper and select what you want to explore. So the member registry will be redesigned completely. You will be able to search for the members, and the entire description that we have in our registry will be searchable. This is an example of an individual members page where you see all the contact information, description, etc. We currently use the Natural Collection Description Standard, but we plan to migrate our registry to the GRSI call registry at GBIF and the Latimer core. But right now it is the Natural Collection Description Standard. This page also will include statistics about the members. If they are core members, which means that they share their sample and specimen data through GGBN, you will see a collection statistics and a portal usage statistics as well. And of course, how many records they share through GGBN. So this is the search page for the main resource, the sample, specimen, and culture collections. And what is important here is that these rows represent GGBN records. So you see immediately for those, there is tissue and specimen available. And you can also expand this. And this is an example for a DNA, tissue, and specimen, where you see the individual parts. And for instance, the availability for loaning could differ, or the repository could differ, etc. So you see all that at once. And since you've heard so much about permits and loans, I wanted to highlight that the permit information is indeed crucial for GGBN because you can request samples for, through the portal. So the good news is that over 73% of all GGBN records have this permit information available, or at least they share the information that it's not available or not required, for instance. And secondly, the loan availability is also derived from the GGBN loan extensions or the Darwin Core disposition term. And of course, this is also important if you have the system in place where you can request the samples. And we really hope and look forward towards the revised version of the permit standard and to implement that in GGBN. So this is an example of the final result of a GGBN record. This is a mock-up, so if you want to look this example up, it will look a bit different because I've added some data just for demonstration purposes. So the first thing you see is the map, of course, but below the loan information. And there will be also a link to the loan policies of the individual institution. And the main data are divided by the material entities. In this case, it's the DNA and the specimen. But there is also a tab for multimedia and sequences, if applicable. And at the bottom, there is permits, if provided, and the dataset description, of course, so that you have contact information. So now I want to focus on the stable identifiers. We want to follow the example of GBIF, DISCO, CTAF, etc., and provide stable, actionable IDs on top of what our providers already provide as GUIDs, for instance. And this will be used for every single material entity, every physical collection object. So every tissue sample, every DNA sample, every specimen that is provided through GGBN. The original institutional GUIDs will stay untouched. There will be an RDF and an HTML representation. And the example how it will look like is id.ggbn.org, and then comes the ID. And you can give it a try. It is a test landing page. Of course, it will be embedded in the new portal, so it looks similar. And this will be launched together with the portal by the end of 2024. And why we do that is to enable the cross-linking between platforms. For instance, ENA, GBIF, DISCO, but also for publication references. And we started a collaboration with ENA to establish cross-references. And already GGBN members provide 1.6 million ENA or NCBI accession numbers through GGBN data. They are associated to the sample data. But through the related material entities, let's say a tissue is derived from a specimen, we can enrich our own GGBN data and create 3.5 million associations between an ENA or NCBI number and the GGBN material entity. And this then will be fed to ENA. And this is a mock-up. This will also be launched as soon as we are ready with our stable identifiers. And if you click on this navigation and cross-references, you will see then the GGBN cross-reference. And this is an example where this sequence is associated to 3 material entities. So originally the link comes from the DNA sample, but because the parent is a tissue and the grandparent is the specimen, they all are at the end related to this sequence. And this is how we can enrich our own data through this relationship, but also platforms like ENA. So I want to thank the entire GGBN portal working group and all the staff members of both GGBN secretariats, ENA in particular, and all the users who gave feedback. Thank you.
All right, thank you very much. Are there any questions about the data portal or the stable identifiers? And Anton's in the audience, so he is happy to answer your questions. I can help. I like it. It's good.
Thank you, and it's a great development, updates. I wonder, or I would like to know the current development or the future plans for any API or a way to access this information automatically. Thank you.
This I can answer, actually. So that's exactly our plan for later this year. So the first step is actually, the very first step was to establish this new GGBN identifiers system, which kind of provides a consistent layer across all data objects handled by GGBN. The second step we are now resting with is the new portal, which is we're pretty close to the launch. And this will be the basis actually also for implementing a redirection mechanism for the new GGBN identifiers. So that if you're using them, you will be redirected either to the human readable splash page in the GGBN portal, or if you come in as a software system, you will be redirected to an RDF representation. And then as a next step, we would like to implement an API with a more efficient way to access all the information in GGBN. So that's exactly our plan. The last part I can't promise for this year, but we are trying hard, and maybe we will finish it this year. Okay.
Don't go away. There's a question online from Esteban Marintes Herrera. How much time did it take to develop the new data portal?
Not so much time, actually. It was really the hard time was the identifier question, to negotiate with all interested partners what the best system would be, how we would deal with the original identifiers provided by the partners and all this. That was quite a steep hill, I would say. But implementing the new portal based on the existing technology, and we're using a lot of existing modules, components provided by the community, like the whole harvesting infrastructure and everything. That's all open source software. It was pretty straightforward, and I hope Gabi will agree because she did most of the work. It was pretty easy, I would say. Are there more questions?
If there's no more questions, we can wrap up early. But I just want to thank all of you for coming. And if you have any friends who have genetic collections, tissues, and DNAs that are interested in joining, we welcome them. And if you have any questions, feel free to reach out to me or Gabby. Yeah, and we're happy to chat. So thank you very much.